Hi, I'm Ashley Voltz with Informa Life Sciences, and today I'm speaking with Dr. Yu Sun, VP of Discovery Product Development with ACROBiosystems. Thank you so much for speaking with me today, Dr. Sun.
Thank you so much.
Let's jump right into it. I had a couple questions for you. First off, what are the most critical attributes of recombinant proteins that impact the success of antibody development in the early stages?
Yeah, as we all know, recombinant proteins are pretty much as the building block for life science research. And specifically for antibody development, people tend to use recombinant proteins as an antigen for immunization and also the potency assays. And there are a lot of very important attributes that we need to consider as the building block for these assays. For example, as an antigen, so we want the proteins to be native-like. So the sequence must be correct. So there shouldn't be any truncations or mutations to really interfere with the nature of the protein. And sometimes the natural proteins actually exist in dimer or trimer. So we want to make the recombinant protein as dimers and trimers as well. Sometimes we do use tags for depurification of the protein. So in that sense, we need to remove the tags from the final product so that the protein is more native-like. So the second attribute I would say is the purity, because we want to have as little contaminations in the final product as possible. A very important aspect that we need to consider is the endotoxin level, because that's really bad for the immune animals, and also it will induce inflammation for the animals, and also induce the bad reaction to the immunization process as well. Also, so these are the two, and of course, the bioactivity is very important as well. So the recombinant protein has to be able to bind to the natural binding partners, for example, the ligand or the receptors as well. Some other very important attributes, I would say, the stability of the proteins. So it has to be not only stable at the storage stage, and it has to be stable during the antibody production stage as well. For example, some proteins can aggregate under certain conditions. So we need to, as a protein maker, we need to make sure our proteins do not aggregate, do not interfere with any assays. So that would be all I can think of right now.
Okay, and how do you balance speed and reproducibility in the development of recombinant proteins to support high throughput screening platforms?
Oh yeah, that's a very good question. So as a company, we have been doing this process, doing this business for 15 years. We were founded in 2010, so literally 15 years from now. We produce on average over 1,000 proteins per year. So we do accumulate a lot of experience and tips in making these proteins. Recently, we actually trained a AI to predict relative expression level based on different sequences of the proteins. We're currently thinking of making it open source to help more researchers in their protein expression field. So as I discussed, so we do accumulate a lot of tips. For example, we can design certain codons, certain expression cassette to make the expression a little bit faster. And also, there are different expression systems that we can utilize to accelerate the process. For example, recently, the cell-free expression systems are quite popular. This is the capability that we have in our company as well. So it can definitely help with the speed. But in terms of the reproducibility, there isn't a trade-off between the two. So whatever we do to accelerate the speed, I would say the reproducibility is the key to keep. So for that, we actually have very stringent QC releasing criteria. For example, as the attribute we just discussed in the question before, we control a lot of factors in terms of the purity, the endotoxin level, the bioactivity, aggregation. So these are all the QC releasing criteria. So we want to make sure that our final product is good for antibody development.
Great. And how do you envision high quality recombinant proteins transforming the antibody discovery and development pipeline in the next 3 to 5 years?
Right, that's a big question. So maybe I'll start with the very basic one. As we discussed before, the antibodies are often, sorry, the proteins are often used as an antigen, right? The better antigen leads to better antibodies. So if we have a high quality proteins, we don't really have to spend so much time on screening all the ineffective antibodies. So that would help with a lot of time saving. And also, I know a lot of companies are using protein libraries to do AI-based de novo antibody designing. So it's completely bypassed the traditional animal immunization stage, right? So that could also help with the novel target discovery. So that's another thing. Also, these proteins are used in validation of the antibodies. So if we use the high quality proteins, you don't really have to spend so much time on tweaking the assays to identify the correct antibodies. Another thing, maybe it's too big to say right now, but I think definitely is the future way, is personalized medicine. So because recombinant protein has a lot of flexibilities, maybe we can tailor the recombinant proteins to target specific genetic mutations to help with certain disease populations that maybe we can design a personalized treatment for these patients. So it's a long way to go, but definitely a way that I would like to imagine.
Wow, that's really interesting stuff. Thank you so much for speaking with me today, Dr. Sun. It was great to hear all about it, and make sure to visit ACROBiosystems next month at Antibody Europe.
Thank you so much.